All right, now we're going to talk about Chapter 7. This is Microbial Nutrition, Ecology, and Growth. So in this chapter, we're going to talk about the process by which nutrients are acquired from the environment to sustain life of the organism. We're going to be dealing primarily with bioelements, and these are the elements that are the basic requirements for life. So things like carbon, oxygen, nitrogen, etc. We have essential nutrients. This is going to be a substance that an organism must get from a source outside its cells. So it cannot synthesize these nutrients, okay? And you've got macronutrients. These are going to be required in large quantities. And micronutrients, also called trace elements. These are required in small amounts. And then we can also talk about nutrients based on what they're composed of. So for example, organic nutrients, these are going to contain carbon and hydrogen, okay? Inorganic nutrients, on the other hand, are going to be any nutrients that have a combination of elements other than carbon and hydrogen. So for organic nutrients, it's going to be like methane, any kind of carbohydrates, lipids, proteins, any biomolecule is going to have carbon in it. And then your inorganic nutrients are going to be things like metals, their salts, gases, and of course water. Now if you took a cell and analyzed its contents, you would find that about 70% of it is water. And if you took the cell and dried it out completely and then analyzed the dry remains, you would find 97% of the dry cell weight is organic in nature. Primarily, it's going to be composed of proteins. And these organic compounds are going to be made up of just 6 elements. You've got carbon, hydrogen, oxygen, phosphorus, sulfur, and nitrogen. So why are these elements in particular significant? Well, carbon is going to form the framework for all biomolecules. It's going to be essential to the structure and function of all organisms and viruses. CO2 is produced by cellular respiration, and we'll learn a little bit more about that in the next chapter. It's also used in photosynthesis. And then you've got carbonate, which is found in the cell walls and skeletons of organisms. Nitrogen, you'll find primarily in the atmosphere, and nitrogen gas is going to be available to microbes that can take it out of the atmosphere and then fix it into other inorganic nitrogen compounds, things like nitrates, nitrites, and ammonium. This is going to be the primary source of nitrogen for algae, plants, and most bacteria. Animals and protozoa, on the other hand, are going to get their nitrogen from organic compounds like amino acids, proteins, and nucleic acids. Usually these are going to originate from other living organisms or their remains. And then all organisms are going to use ammonia to synthesize amino acids and nucleic acids. Oxygen. Oxygen is going to be necessary to metabolize nutrients. You'll learn more about this next chapter, but it's the final electron acceptor in oxidative phosphorylation. This is where ATP is made. It's also going to be a significant element in organic and inorganic compounds. Hydrogen is one of the main constituents of water. Hydrogen gas, hydrogen sulfide, and methane gases are going to be produced and used by bacteria and archaea. And then you've got hydrogen ions that are going to be the basis for transfers of cellular energy. They're also going to help establish and maintain the pH of cells. Phosphorus is significant. It's a component of DNA and RNA. It's also going to be found in ATP, so that's going to be your cellular energy. And NAD, again, helps with energy reactions. And then, of course, it's going to be present in those phospholipids, which form that phospholipid bilayer of the cell membrane. Sulfur is going to be used by some bacteria as an energy source. It's also found in vitamins such as B1. And then you've got sulfur in certain amino acids that where they form bonds with one another, and this helps shape and stabilize the proteins. Okay, and then we've got some other nutrients and their roles. So you've got potassium, sodium, calcium, magnesium, chloride. You'll notice a lot of these are actually electrolytes in our body, but they play crucial roles in microorganisms as well. You've got zinc. This is going to be an enzyme cofactor, so it helps enzymes function. Iron, important with respiratory proteins. And then several micronutrients, okay? And these are required in tiny amounts. They usually serve as cofactors for enzymes, so they help with enzyme function. Okay, so where an organism gets its carbon is going to divide organisms into two types, okay? So you've got heterotrophs and autotrophs. Heterotrophs are going to obtain their carbon in an organic form, so proteins, carbohydrates, lipids, nucleic acids, and they're going to obtain it through other living organisms. Autotrophs, on the other hand, are going to use inorganic carbon, so CO2, and they do not need to obtain that from other living things. Okay, so we are heterotrophs, just to give an example. And an example of an autotroph would be something like a plant that takes its CO2 from the atmosphere. Growth factors you need to know for your test. These are going to be organic compounds that cannot be synthesized by an organism. Okay, they lack the mechanisms to synthesize these growth factors. So it has to be provided as a nutrient from outside of the organism. Examples here would be essential amino acids and vitamins. Nutritional types. So I want you to know this for the exam as well. You can kind of break nutritional type into two categories depending on carbon source or energy source. Okay, so if you're looking at carbon source, that's where we talk about our heterotrophs versus our autotrophs. Okay, where do they get their carbon? Heterotrophs get it from other organisms. Autotrophs use CO2. But you can also look at the energy source. Okay, so energy source being chemotrophs, where they gain their energy from chemical compounds, or phototrophs, where they gain their energy through photosynthesis. All right, so this chart is just showing nutritional categories of microbes, and it's showing by energy and carbon source, okay? So here we have autotrophs, and then here onwards we have heterotrophs. So we've got two types of autotrophs. You have the photoautotroph. This energy source is going to be sunlight. And you have the chemoautotroph. The energy source is going to be simple inorganic chemicals. Don't worry too much about chemoautotrophs. I want you to focus more on photoautotrophs and then the differences in the heterotrophs, okay? Your chemoheterotrophs, these guys are going to use nutrients from other organisms. So this is going to be protozoa, fungi, a lot of different bacteria, and animals. And you've got a couple of categories here. You've got saprobes, which are going to metabolize the organic matter from dead organisms. You need to know that for the exam. And you've got symbiotic microbes. These guys obtain organic matter from living organisms, okay? So examples there would be parasites, commensals, and mutualistic microbes. We'll talk more about these guys here in a little bit. Photoheterotrophs, don't worry too much about that. This is going to be your purple and green photosynthetic bacteria. Photoautotrophs, we can talk about them being oxygenic or anoxygenic. All this is saying is, do they produce oxygen? We usually think of photoautotrophs as being oxygenic, right? So they take CO2 and then they release oxygen. However, there is anoxygenic photosynthesis. You'll see this with purple and green sulfur bacteria, where they produce sulfur instead of oxygen. Chemoautotrophs, you don't have to worry too much about this for the exam, but I do want you to be aware that these guys can survive totally on inorganic substances. And an example here would be the methanogens, okay? These are archaea. and they produce methane gas under anaerobic conditions, so conditions without oxygen. They're going to derive their nutrients from CO2 and hydrogen gas, and they're going to thrive in environments that lack oxygen. Prime example here being the guts of cows. These guys are a key player in the digestive process of ruminants. They help break down food, but they also release methane. Okay, more on heterotrophs, and we've got some more definitions here. You've got chemoorganotrophs. So these guys are going to derive carbon and energy from organic compounds. For example, starches, glucose, things like that, okay? This is going to be aerobic respiration. We'll talk more about that next chapter in detail. The two categories I want you to be aware of with heterotrophs are going to be the saprobes and the symbionts, okay? Saprobes, these guys feed on dead matter. They can be opportunistic pathogens, okay? Symbionts, on the other hand, are going to derive their nutrients from the host itself. They can obtain nutrients from living or non-living sources, and some of these are going to be obligate parasites. Okay, so for the exam, you're going to want to probably study this chart and be familiar with these definitions. I would definitely know the difference between an autotroph and a heterotroph, and a photoautotroph and a chemoheterotroph in particular. And I would know what a saprobe is, and I would definitely go over the symbiotic microbes, which we'll talk about here shortly, okay? Okay, so how do these organisms move molecules in and out of their cell? How do they obtain nutrients to begin with? Well, they have to have some form or some method to transport them. So we're going to talk about two main types of transport. You've got passive transport and active transport, okay? Passive transport does not require any energy expenditure by the cell. It just happens. Whereas active transport, the cell is going to have to expend energy to obtain whatever molecule they're trying to transport across their membrane, okay? So in passive transport, we said this is not going to require energy. And the reason for this is because the substances are going to exist in a gradient. That means there's going to be a higher concentration on one side of the membrane and a lower concentration on the other side of the membrane. Substances are going to move from that area of high concentration towards the area of low concentration. And there's three methods. There's simple diffusion. This is what you see when you mix sugar and water. Simple diffusion. You've got osmosis. This is the diffusion of water specifically, okay? And then you've got facilitated diffusion. Solutes are going to move down their concentration gradient, right? They're going to go from areas of high concentration to low concentration, but they do require some sort of carrier, like a protein, that helps move them across the membrane. Compare that to active transport. So active transport is going to require energy. It uses proteins, carrier proteins, right, to carry these molecules across. And it's going to move these molecules against their gradient. So using active transport, you can move molecules from areas of low concentration to areas of high concentration against their gradient. Three examples here. We have carrier-mediated active transport. This is going to use proteins to move those molecules across the cell membrane. You have group translocation. This also uses proteins to move molecules across the membrane, but as they are moving across the membrane, they are chemically altered. And then you've got bulk transport. This is going to be your endocytosis, exocytosis, and penocytosis. Okay, this is an example of simple diffusion, where there's going to be a net movement of molecules down their concentration gradient, right? So from areas of high concentration, for example, this region here of the orange, where there's a lot of dye here, and then so it's going to move from this high concentration out to areas of low concentration. You can see on the periphery here, you have just plain water. Okay, so here it's spreading out until it's uniformly orange. throughout the solution. And the reason for this is random thermal movement. This is also called Brownian motion. And all that means is that these molecules are just moving randomly, and they will continue moving randomly. And that random motion causes them to spread out uniformly, okay? Okay, a little bit about osmosis. So cell membranes are going to be selectively permeable. So they're going to allow some molecules to enter and block other molecules as well. Okay. Generally, they're going to allow the free diffusion of water. Now, when concentrations of the solutions differ on either side of that selectively permeable membrane, one side is going to experience a loss of water, and the other side is going to gain that water until solutions on both sides of the membrane are equal. So water is going to move from the side with the lower concentration of solutes, or the higher amount of water, the higher concentration of water, to the side with the lower concentration of water. And I'll show you what I mean. All right, here in this beaker we have a semi-permeable membrane that's going to allow water to flow through, but it's going to block these solutes from moving, okay? Now you can tell on the left side here, there is relatively more water per solute, right? Than over here, there's less water per solute. So what's going to happen is water is going to move from areas of high water concentration, so a lot of water, to areas of low water concentration. Another way to think about this is that water is going to move from areas of low solute concentration to areas of high solute concentration until the solutions on both sides of the membrane are equal. Okay, tonicity. Tonicity is going to be the relative concentration of solutes in a solution compared to another solution. And tonicity is going to influence the movement of water across a semi-permeable membrane. You've got hypotonic, isotonic, and hypertonic, okay? So hypotonic is going to be a solution that has a lower concentration of solutes compared to another solution. For example, when you place a cell in a hypotonic solution, water is going to move into the cell. So there's less solutes outside of the cell than inside of the cell. So water is going to move from that area where there's fewer solutes to the area with more solutes. So it's going to move from outside the cell to inside the cell, which can cause the cell to swell, potentially bursting the cell, okay? An isotonic solution is going to be a solution that has the same concentration of solutes as another solution. There's going to be no net movement of water in an isotonic solution, and the cell is going to remain the same size. On the other hand, there's hypertonic solution. This is going to be a solution that has a higher concentration of solutes compared to another solution. So when you take a cell and you place it in a hypertonic solution, there's going to be more solutes outside of the cell than inside of the cell. So water is going to move from inside the cell out of the cell and causing the cell to shrink. So let's look at a few examples. Here we're looking at cells with cell walls. So this would be something like a bacterium. So let's say you place a bacterium in an isotonic solution. So you've taken this bacterium and you've placed it in a solution that has the same amount of, the same concentration of solutes as inside of the bacterium. There's going to be no net movement of water. Let's say you take the bacterium and you place it in a hypotonic solution instead. So the solute concentration inside of the cell is higher than the solute concentration outside of the cell. Well, water is going to diffuse into the cell, and that's going to cause the cell to swell. Now, in animal cells, usually this will, if you have enough influx of water, can cause the cell to burst. However, we're looking at cells with cell walls here, so in this example, the cell wall is going to help prevent this cell from bursting. Next, you have a hypertonic solution. This is going to be where the solution outside of the cell has a higher solute concentration than the solution inside of the cell. So water is going to diffuse out of the cell here. This is going to cause the cell to shrink, and this is known as plasmolysis. Okay, here you can see the examples in cells without cell walls. So this here is an isotonic solution. There's going to be no net movement of water in or out of the cell. Now, if you took that same cell and placed it in a hypotonic solution, remember, water is going to go into the cell in a hypotonic solution. And notice this is a cell without cell walls. So it is going to swell until it bursts if there's no mechanism to remove that water. On the other hand, if you take this cell and you place it in a hypertonic solution, you're going to see the cell shrink. This is because the water leaves the cell and goes into the hypertonic environment. Okay, and then finally in passive transport we have facilitated diffusion. So facilitated diffusion is going to use a special carrier protein in the membrane that is going to bind a specific substance and move it across the membrane down its concentration gradient. Remember, passive diffusion or passive transport, is always going to be down the concentration gradient. These carrier proteins are going to have specificity. That is, they're going to bind and transport only one type of molecule. For example, glucose. Facilitated diffusion is going to exhibit saturation, and what that means is that how fast these cells can transport these molecules depends on the number of binding sites on the transport proteins. When all of those binding sites are taken up, you can't transport more molecules than you already are, so your rate of transport is limited. Here's an example of facilitated diffusion. So you have a high concentration of solutes on the outside of the cell, and then a low concentration of solutes on the inside of the cell. Here's your carrier protein. Here's a solute that goes and it connects with the binding site of the carrier protein, and then the carrier protein facilitates the movement of that solute across the membrane. Remember, this is going down the concentration gradient, so there's no energy required. Okay, on to active transport. Carrier-mediated active transport. Now remember, this is going to require energy. You have permeases and pumps. These are specific membrane-bound transporter proteins that are going to move solutes against their concentration gradient. And they're going to utilize ATP in order to do this. All right, so here you have the solute that's outside of the cell. Here's the transporter protein. These are the ATP binding sites on the transporter protein. And you'll notice solutes are usually bound in some sort of protein, a solute binding protein. And what happens is this solute binding protein connects with the transporter protein. And ATP is activated, and when that happens, it opens this channel up and allows the solute to move through. And remember, the solute is going to be moving against its concentration gradient, so it's going to be going from areas of low concentration to areas of high concentration. Group translocation is going to be a lot like carrier-mediated active transport, okay? But during transport, the molecule is going to be chemically altered or activated. And the whole point of this is that it helps save a little bit of energy for the cell. This is going to be primarily used by bacteria and archaea. Okay, so here you have your transport protein. Here are the molecules that are being transported. And you can see as they're transported through, they are altered chemically, and now they are activated and ready for use in the cell. And all this does is it saves a step for the cell and it helps it conserve energy. All right, and finally in active transport, we're going to talk about endocytosis. This is going to be bringing substances into the cell through a vesicle or phagosome. You have two types of endocytosis. There's phagocytosis and pinocytosis. Phagocytosis is so called because it's, quote, cellular eating. And what happens is the cell ingests substances or even other cells, usually using pseudopods, okay? Pinocytosis is cellular drinking, and this is where the cell ingests fluids and or other dissolved substances. So on the left is an example of phagocytosis. You have the extracellular area here, and you can see a food particle that the cell wants to take in. So the cell forms these pseudopods, or false feet, that kind of wrap around the food particle and bring it in the form of a food vacuole. Over here you have pinocytosis. You can see the dissolved solutes that the cell wants to bring in. And so it kind of just creates an invagination in the cell membrane, brings that fluid in, and then kind of pinches off, forming a vesicle. All right, I think this is a pretty useful chart for you to study when you're getting ready for the exam. So things I want you to know the difference between passive transport and active transport. Remember, passive transport is going to move substances down their concentration gradient, and it's not going to require energy. Active transport is going to require energy, and it's going to move molecules against their concentration gradient. I want you to know osmosis, or the diffusion of water. I want you to know carrier-mediated active transport. And I want you to know the different methods of bulk transport. You need to know the difference between phagocytosis and pinocytosis. Okay, so question here. If a cell is in a concentrated glucose solution and the glucose is moving into the cell through a carrier protein down its concentration gradient without expending energy, this would be an example of what? Well, everything here is saying passive transport, okay? It's moving into the cell down its concentration gradient without energy expenditure. And it's using a carrier protein. So we know it's not simple diffusion. Simple diffusion does not require a carrier protein. So it must be facilitated diffusion. Okay, environmental factors that influence microbes. So I want to introduce the concept of the niche. Your book uses a slightly different definition for this, but the definition I want you to know is that a niche is the specific environment or role that an organism occupies within an ecosystem. And this is going to include its interaction with other organisms, its interactions with its habitat, and the resources it uses, okay? Environmental factors in the niche are going to affect the function of the microbes. It's going to affect how their enzymes work, okay? So these factors are going to include temperature, oxygen, pH, osmotic pressure, and barometric pressure as well. Okay, so temperature and microbes, we talk about minimum, maximum, and optimum temperatures. Minimum temperature is going to be the lowest temperature that allows the microbe to grow. The maximum temperature is going to be the highest temperature that allows the microbe to grow. Now, the optimum temperature, on the other hand, is going to promote the fastest rate of growth of the organism. More definitions here. You have your psychrophiles. These are organisms that grow with an optimum temperature below 15 degrees Celsius. They're capable of growth at freezing, okay? Then you have your mesophiles. Optimum temperature here is going to be between 20 to 40 Celsius. Body temperature falls in this range, so you'll see most human pathogens are going to be mesophiles. Then you have thermophiles. Their optimum temperature is going to be hot, higher than 45 Celsius. Oxygen use. Not all microbes can use oxygen. In fact, some die in the presence of oxygen. The reason for this is because when you utilize oxygen, it's going to be transformed into different toxic products, okay? Reactive oxygen species, peroxide, superoxide ion, and singlet oxygen. The thing with these is that they tend to have high energy electrons. and these electrons are highly reactive and can damage cells and tissues. Most cells have developed mechanisms that neutralize these toxic byproducts of oxygen. For example, catalase. We'll learn a little bit about catalase in this course. But what catalase does is it converts toxic hydrogen peroxide into water and oxygen. Now, if a microbe is not capable of dealing with these toxic oxygen byproducts, it's going to be forced to live somewhere where there is no oxygen. Now, when we talk about microorganisms, especially bacteria, one very common way to classify them is whether they can metabolize oxygen. So you have aerobes. These guys can utilize oxygen and detoxify it. And you have anaerobes, which cannot utilize oxygen. So for aerobes, you have obligate aerobes. These guys have to have oxygen. They cannot grow without it. You have facultative anaerobes, and these guys can use oxygen, but they can also grow in its absence. So you'll see facultative organisms can survive in a wider range of environments. Anaerobes, you have an obligate anaerobe. These guys cannot detoxify oxygen, so they have to live in an oxygen-free environment. Then you've got aerotolerant anaerobes. These guys don't utilize oxygen, but they can survive and grow in its presence. Growing anaerobes, strictly anaerobic, obligate anaerobic bacteria is going to require special media, special methods of incubation, and special handling chambers. So we are not going to be utilizing anaerobes in this course. Most of the microbes that we're going to use are going to be facultative anaerobes. And then we have some obligate aerobes that we use as well. Most microbes are going to require some CO2 in their metabolism. And here's another definition, the capnophile. These guys grow best at higher CO2 pressures than normally present in the atmosphere. Most microorganisms are going to grow at a pH between 6 and 8. Neutrophiles, that's going to be these guys growing within a range of about 5.5 to 8. Remember that neutral on the pH scale lands right at 7. You've got acidophiles. These guys are going to grow at extremely acidic pHs, so low pH numbers. And you've got alkalinophiles. These guys are going to grow at extreme alkaline pH or high pH numbers, okay? So osmotic pressure, most microbes are going to live in hypotonic or isotonic environments. Osmophiles, these guys prefer environments with higher osmotic pressure, and specifically halophiles require a high concentration of salt. You have obligate halophiles, which are going to grow optimally in solutions of 25% sodium chloride, but they're going to require at least 9% sodium chloride, and this is going to be environments like salt lakes and ponds. You have osmotolerant microbes. These guys do not require a high concentration of solute. For example, facultative halophiles. These guys are remarkably resistant to salt, and one that we'll use in this course is going to be Staphylococcus aureus. We use a special growth medium called mannitol salt agar, and this guy is selective to isolate and identify Staphylococcus aureus. Because it has a high amount of salt in it that most microorganisms cannot tolerate. Staph, on the other hand, is very resistant to high salt concentrations. Barophiles, these guys can survive under extreme pressure, for example, deep in the ocean, and they will rupture if exposed to normal atmospheric pressure. And water plays a role as well, okay? So only dormant cell stages are going to tolerate extreme drying. And these dormant cell stages are going to be your endospores, remember bacterial endospores, and cysts, okay? Cysts like protozoan cysts. These guys are dehydrated cell stages, and the way they are adapted is to survive extreme drying. All right, so how do these microorganisms interact with each other? We've got two different kinds of microbial associations. We have symbiotic and non-symbiotic. So symbiotic relationships, this is going to be when organisms live in close nutritional relationships. And this is going to be required by one or both members to live. So we have mutualism, commensalism, and parasitism. I want you to be familiar with all of these terms. Mutualism is where both organisms benefit. Commensalism is where the commensal organism benefits and the other organism is not harmed. Then you have parasitism. This is where the parasite benefits and the host is harmed. Then you have non-symbiotic associations, and this is going to be where the organisms are free living and their relationships with one another are not required for survival. So remember, symbiotic relationships are required for survival in symbiotic organisms. So our non-symbiotic associations, we have syntrophy. This is where the members cooperate and share nutrients with one another, but again, remember they don't need one another for survival. Then you have amensalism. This is where a microorganism inhibits or destroys another organism. Okay, our symbiotic relationships. Let's start with mutualism. This is where both members benefit in the relationship. There's obligate mutualism. These guys require one another to survive. Then there's non-obligate mutualism. These guys can be separated and they can live apart. And we'll talk about, for obligates, we'll talk about how termites rely on trichonympha. And we'll talk, for non-obligates, we'll talk about how Euploides helps out unicellular green algae. In commensalism, the commensal organism is the one who benefits and the other organism is neither harmed nor benefited. We'll talk about Haemophilus and Staphylococcus. And then there's parasitism. The parasite depends on the host and benefits. The host is actually harmed. And we'll mention Rickettsia and Chlamydia here. Okay, so termites require or need trichonympha, which is a type of protozoan, in order to help them break down wood. So they would not be able to digest the wood that they eat if not for these endosymbiotic protozoa. And protozoa break down the cellulose by secreting enzymes that digest it, and this releases simpler sugars that then the termites can absorb and use as an energy source. So without these protozoa, termites would starve. This is an obligate mutualistic association. So trichonympha needs the termites as well in order to live. It cannot live and reproduce outside of the termite's body. So again, obligate mutualism here. Here we have Euploides, which is another protozoan, and green algae. And what Euploides does is it provides a safe environment for the algae. The algae in turn can perform photosynthesis, producing oxygen and glucose that benefits Euploides. Both organisms benefit in this relationship, but it's not obligate. They don't need one another to survive. Next, we have commensalism. Remember, this is where one organism benefits and the other organism is neither harmed nor benefited. We have, for an example here, Haemophilus and Staphylococcus. Haemophilus is a bacterium that, among other things, can colonize the respiratory tract, can be a normal member of the respiratory tract, the upper respiratory tract. Staphylococcus is also often a colonizer of the upper respiratory tract, and Staph epidermidis is a colonizer of the skin. These are all normal bacteria to find on the body. Now what happens is Haemophilus needs certain nutrients like hemin to survive, and these nutrients are not readily available in the environment. Staphylococcus produces these nutrients, so Haemophilus kind of hangs around Staphylococcus and uses those nutrients to its benefit, but it does not harm Staphylococcus. And here you can see this streak colonial growth here. This is a staph colony. And then you can see these small satellite colonies popping up on the periphery, and these are Haemophilus bacteria. Okay, parasitism. So here we have malaria as the example, and this is going to show multi-level parasitism. Mosquitoes are parasites of humans that carry their own parasites, and their own parasites are the malaria parasites, Plasmodium. Plasmodium is a protozoan that can also infect us. And Plasmodium essentially lives its life cycle between the mosquito and us, and utilizes us to live. So it is a parasite. It needs a host in order to survive. And just a little bit more about parasitism. So we have ectoparasites and endoparasites. Ectoparasites are going to live on or feed on the outside of the host body. This is going to be things like ticks, fleas, mosquitoes, and these guys are often vectors of other parasites. So they're usually viruses, sometimes protozoa, that they can spread. Endoparasites, on the other hand, are going to live on or feed on the inside of the host body. This is going to be things like protozoa. So we'll talk about malaria. We'll talk about toxoplasmosis. We'll talk about giardiasis. Helminths, or parasitic worms, we'll also talk about in this course. Viruses we'll talk about. Remember, these guys are obligate intracellular parasites. And some bacteria are also obligate intracellular parasites. For example, Rickettsia and Chlamydia. Okay, non-symbiotic associations are going to occur when the organisms are free living and they share a habitat. So we have syntrophy, or cross-feeding. This is where the microbes sharing the habitat feed off of substances released by the other organism. I'll show you an example here in just a second. Then you have amensalism. This is where one member of the association produces a substance that either harms or kills the other one. This can also be called antibiosis. And the example we'll talk about will be mold that produces penicillin, which kills nearby bacteria. And the goal of this is that the mold, penicillium mold, kills the bacteria in order to decrease competition on the resources in that shared habitat. All right, here is an example of syntrophy. Remember, this is going to be cross-feeding. So we have the nitrogen fixer Azotobacter here. It's going to feed Cellulomonas ammonium. So Cellulomonas is going to take that ammonium and feed off of it, and in return, it's going to produce glucose. And Azotobacter is going to feed off the glucose. Here's our example of amensalism. So you have a penicillium colony. This is mold, and it produces penicillin. And then you have a bacterial colony. Here you can see on the streak on the plate. And here you can see an area of clearing around that penicillium mold where the bacteria have been killed. Okay, another really important microbial relationship we need to talk about is biofilms. So biofilms result when organisms attach to a surface, and they use an extracellular matrix that binds them together to this surface in complex organized layers. They actually begin to communicate and cooperate with one another in this biofilm, and this is called quorum sensing. So how do biofilms form? Well, first, you're going to have free-swimming cells lose their motility, and they're going to settle down onto a surface. Next, the bacteria are going to produce an adhesive matrix, right, that extracellular matrix around it, and this is really sticky. The cells are then going to start secreting inducer molecules, and if you look at one cell close up, you can see what the inducer molecule does. It essentially turns on a gene in the bacterial chromosome to start creating a certain protein, usually enzymes, okay? And then what's going to happen is these cells are going to secrete their enzymes in unison to digest these food particles. Okay, so just a little bit about our relationship with microorganisms. Our body is going to be a rich habitat for symbiotic bacteria, fungi, and even some protozoa. These organisms all together account for more, there's more organisms, microorganisms on and in our bodies than there are human cells. Okay, and together these microorganisms are called the normal microbial flora, also known as the microbiota, also known as the human microbiome. Okay, all of those terms are interchangeable. You'll see a variety of relationships between us and our microbiome. Some organisms have a commensal relationship, a parasitic relationship, and even synergistic relationship. And these guys affect our physiology, our immunity, and even our genetics. One very important aspect of our microbiome is our gut flora. Okay, so we have a lot of microorganisms that take up residence in our intestines. And these guys help prevent disease in two ways. Number one, they take up physical space, so there's less attachment sites for pathogens to latch onto and set up shop. Number two, they secrete molecules that are inhibitory to pathogens. So the microbial flora in the gut are incredibly important to preventing disease. Microbial growth. So when we talk about microbial growth, we're going to be talking about it at two different levels. There's growth at the cellular level, where the cell itself increases in size. And then there's growth at the population level, where the number of cells increase. Now, division of bacterial cells is going to occur mainly through binary fission. What happens here is the parent cell enlarges, it copies its DNA, and then it's going to divide itself into two daughter cells, each identical to one another. Here you have binary fission, so you have the cell at the beginning of the cell cycle. Here the cell has enlarged and it's copying its chromosome. Here we have two copies of the chromosome, and you can see the cell is starting to divide. Here you've got the cell is almost completely finished dividing, and then finally you have two identical daughter cells that are the result of this binary fission. Population growth. So the time that's required for a complete fission cycle to occur is called the generation time or the doubling time. So what happens is each time that binary fission cycle, each time the organism undergoes that binary fission cycle, it's going to increase the population by a factor of 2. In other words, it doubles itself, okay? This is exponential growth. Generation times are going to vary on the organism. Some organisms can double themselves within minutes, and some take days. But generally speaking, bacteria have very rapid generation times. And then this I just put in here to show you how the population grows exponentially. So you can see here we begin with one cell, and then after 1, 2, 3, 4, 5 generation times, you have 32 cells.